So as DePion mentioned, I am joining from Medical College of Wisconsin. So this is a skyscape image of the lakefront of Milwaukee. So not to be confused with the University of Wisconsin, which is in Madison. So I've already had to clarify that a couple times this morning. So this is a cardiovascular seminar, so I probably don't really need to spend too much time on this first slide. But what my lab is really interested in is understanding how the mammalian heart can repair after an injury, such as a myocardial infarction. So we know that the adult mammalian heart is poorly regenerative. So this is just an image of a mouse heart before infarction and after infarction. And so what we all can clearly appreciate is that that infarct region results in this loss of functional myocardial tissue and this development of a fibrotic scar, and in many cases, progression to heart failure if individuals experience a heart attack. So the therapies for progression to heart failure are limited. So we're really interested in understanding the various cell types and molecular mechanisms by which we can promote healing in the mouse heart and apply those to human therapeutics. So there are a multitude of cell types that are involved in repair processes and regenerative processes. So one of the reasons that the adult mammalian heart fails to regenerate, unlike some tissues and organs such as skin or liver, which are much more regenerative, is in part because of this terminally differentiated state and non-proliferative state of cardiomyocytes. So one major area of interest is how we can remuscularize the tissue, whether it be from driving proliferation of pre-existing cardiomyocytes, remuscularizing via delivery of stem cells that can be differentiated into cardiomyocytes that can contribute to the contractile properties of the heart, or mechanisms such as transdifferentiation from fibroblasts to cardiomyocytes. So there's a whole field of individuals working on various aspects of remuscularization. But to achieve comprehensive and functional heart repair, we not only need to achieve remuscularization, but we need to understand how those cardiomyocytes can regain this electromechanical coupling. We need to understand how this rigid fibrotic scar can be degraded or resolved without causing a catastrophic rupture event. We need to understand how the heart can be, this infarct region can be perfused. And we're learning a lot more about how immune cells are involved in this process. So my lab studies various aspects of particularly remuscularization, resolution of fibrosis. We do some of this electromechanical coupling, and we're interested in this immunological balance. So today I'm going to be talking about several different cell types that are influenced by interleukin 13 in heart regeneration and repair. Yep.
So the remuscularization, is that interchangeably, do you interchange that with revascularization, for example? Or does that fall under the same topic?
Sorry, what's the question?
Revascularization
right.
versus revascularization.
Right. So I consider them to be distinct processes, both of which would be required.
Okay, required.
Yeah, yes.
Okay. So then what do you think the timeline is? You have to go from remuscularization to angiogenesis / fibrogenesis. Does that have to be complete before one step has to be complete before you go to the next one, or it doesn't really matter?
So it probably matters. I don't know that I have an answer to that question. Yeah. But from the literature, driving remuscularization, there's probably some signals from those newly muscularized cells that then would promote the angiogenesis. Yeah, right. So I think that's still an open question, right, for sure. Mm-hmm. So unlike the adult mammalian heart, there are certain examples of organisms that do very efficiently regenerate their hearts. For example, zebrafish throughout adulthood can regenerate their hearts. So if you clip off about 15% of a zebrafish heart, within about 30 to 60 days, that heart apex will grow back. In 2011, Hesham Sadek and Eric Olson's group published that neonatal mice are also capable of fully regenerating their hearts. So if you take a newborn, 1-day-old mouse and either give them a little heart attack or clip off a small piece of their heart, within about 3 to 4 weeks, the heart will fully regenerate. Now, what my lab is interested in is applying these pro-regenerative models and really understanding how this process successfully works in these models so that we can reactivate them in non-regenerative scenarios like adult mammals post-infarction. Now, natural examples of heart regeneration, such as in zebrafish or in neonatal mice, was first, one of the cell types that was really of interest is the cardiomyocyte. And that's because these models that are pro-regenerative also happen to have proliferative competent cardiomyocytes. So in mice, for example, within the first few days of life, cardiac growth is achieved primarily or in large part by hyperplasia of pre-existing cardiomyocytes. So by about day 4 to 5, the proliferative growth switches over to hypertrophic growth. So in a 7-day-old or adult heart, the cardiomyocytes are very resistant to proliferation. So when I was a postdoc, so this is the example that I was alluding to of like how we first became interested in this interleukin signaling in the first place. When I was a postdoc in Rich Lee's lab, I started in his lab in 2011, and sorry, I'm also just going to grab my phone because I don't see a clock in here, and I have behind me. Okay, so well, I'll just put it there just to make sure I don't go till 1:30 here. So when I started in Rich's lab in 2011, this neonatal mouse heart regeneration model had just been published. And we were really interested in understanding, the assignment I was tasked with was define cardiomyocyte dedifferentiation. That was my project, which is like a sufficiently vague assignment for a postdoc. And so the way I sort of thought about that is, what are the factors that are potentially regulating the proliferative state of cardiomyocytes? Okay, we're trying to understand what factors could trigger this. So to summarize my entire postdoc in one slide, which was I guess there was like an editorial on this paper in 2015, was we basically took several different models of cardiomyocytes as they transitioned from a proliferative state to a less proliferative state and vice versa. So we modeled this in a bunch of different ways. So we took embryonic stem cells and differentiated them into cardiomyocytes. As they differentiate, they lose proliferative potential. The same is true with just in vivo maturation. As I mentioned, newborn hearts the myocytes are very proliferative, and as they age, that decreases. And then on the other hand, we looked for models whereby myocyte proliferation increases. So if you snip the apex of a newborn mouse heart, the cardiomyocytes increase their proliferative incidence. So it's a model of increased proliferation. And if you take adult mammalian cardiomyocytes and culture them, they'll upregulate proliferation genes. So this is kind of our in vitro model of an increase in proliferation. So we basically did RNA-seq at several different time points from all of these different models and performed gene network analysis and upstream regulator analysis to identify factors that could regulate that process bioinformatically. And what we found from all of this we found several candidates, some of which had been also demonstrated to be pro-proliferative in recent literature. But one unique factor we found was interleukin-13. So interleukin-13 was predicted to be an upstream regulator of myocyte proliferation. And so that was pretty much the end of this paper, and now my transition to independent faculty, when I started seeing if this were true or not in vivo. So I started my lab in 2016. I moved back to Milwaukee, which is, as mentioned, where I'm from. And the first goal was to see, okay, does interleukin-13 do anything with heart regeneration at all? So at the time, we had no evidence. So the first experiments that we did was we obtained IL-13 global knockout mice, did a very simple experiment where we performed apical resection in newborns and did comprehensive physiology and cell histological phenotyping, and indeed found that interleukin-13 knockout mice do have this impaired heart regeneration. So if we look at the hearts at 21 days post-injury, you can appreciate from these trichrome stains that the interleukin-13 mice fail to completely resolve this scar, as indicated by this increased area of apical fibrosis. And that was also correlated with increased ventricular dilation. So at a functional and histological level, they failed to fully regenerate. Now that's great, but the next question is how is it doing this? Is it via IL-13 directly signaling to myocytes to mediate a proliferative response? That was our initial hypothesis. So we next moved on to dissect the cell signaling mechanisms of IL-13 in neonatal heart regeneration. So I mentioned that the global IL-13 knockout shows impaired heart regeneration. And then we created a series of receptor deletion models. So interleukin-13 signals through this type 2 receptor, which is comprised of IL-4RA and IL-13RA1 receptor subunits. And it's important to note that IL-4RA can also heterodimerize with this gamma chain, which is only responsive to IL-4. So we generated conditional knockout mice by deleting this IL-4RA subunit. So we tried to make floxed mice with the IL-13RA1 subunit, but they never went germline, so we had trouble getting that line. So for some of these neonatal experiments, the reason I say my title is interleukin signaling and not specifically 13 is because we can't always distinguish between IL-4 and IL-13 in some experiments, but later on you'll see how we start to tease those apart. So we demonstrated that deletion of IL-4RA really recapitulated the phenotype of IL-13 knockout. They also failed to regenerate. And sure enough, when we deleted the receptor in cardiomyocytes, we also got a regeneration deficit. However, this regeneration deficit, when we deleted the receptor only in myocytes, did not fully, it was more mild. It didn't fully recapitulate the cytokine or the receptor deletion, suggesting that there were other cell types involved. So IL-13 receptor is expressed on a lot of different non-hematopoietic cell types, as well as immune cells such as like macrophages and neutrophils. And for several reasons, we were interested in the role of its signaling in macrophages. And we deleted the IL-4 receptor in macrophages and again found that these animals also showed this regeneration deficit. So from this several studies, we really demonstrated that IL-13 signaling and or IL-4 signaling, in some cases we can't discriminate, does mediate on this natural regenerative process, at least in neonatal mice. So what happens with these heart regeneration phenotypes is that a lot of these really important factors that mediate development also mediate regeneration. And a lot of them decline into adulthood. So for example, YAP activity in cardiomyocytes is extremely pro-proliferative. It mediates proliferation during development and is also very important in heart regeneration. And when it's reactivated in adults, you can actually drive this cardiomyocyte proliferative event. So we asked if the same is true about IL-13 and IL-4. Is this something that's enriched in neonates that we can then re-engage in adults to promote cardiac repair? So we measured expression of the receptors in the heart. We didn't really see much of a difference between neonates and adults. We did serum ELISAs for these cytokines. We didn't see much of a difference. So we really then wanted to get to the source of the cells that are producing these cytokines. And if for those of you that work with cytokines, it can be very challenging to detect them. They're abundant, they're expressed at very low abundant levels, and it's hard to identify the cellular source. So we use these two reporter lines whereby for IL-13, where YFP, an iris YFP is inserted into the 3' UTR of the gene. So basically any cell that's expressing IL-13 will be YFP positive. So it's not like YFP is like fused with IL-13. It's there's an iris element in there. So the cells expressing IL-13 will be YFP positive, and we can therefore detect them by flow cytometry. We also did this for IL-4 because of the overlapping roles of these cytokines. This model has iris GFP, but similar concept. And we basically measured expression in neonatal and adult hearts. And we looked at all sorts of different cells that are known to express IL-13 and IL-4, including T cells, B cells, basophils. And we really found predominant expression of both IL-13 and IL-4 in these ILC2s, which are these innate lymphoid cell types. And so you can appreciate that both YFP and GFP are much more highly expressed in neonates, which are in blue, in neonatal ILC2s compared to adult, in both the injured and following MI. So this demonstrates that IL-13 and IL-4 expression in the heart, at least the only source of, or the strongest source of expression that we could find, declines tremendously from neonatal to adult stages. So not only does expression of IL-4 and IL-13 within ILC2 cells decrease into adulthood, but so does the frequency of this cell type in general. So in the neonatal heart, compared to like in the uninjured setting, the frequency of ILC2s in the heart is fairly similar. But following an injury like an MI, neonates mount a stronger likely proliferation of ILC2s compared to adults. So the number of the cell type and the expression of IL-4 and IL-13 is much stronger in neonatal hearts.
Mm-hmm. Have you investigated if this effect is known as staying only to the heart, or do you think of
So that's an excellent question, and we haven't looked at any other organs besides the heart. Mm-hmm. That's a great question. So considering there's a much stronger abundance of this protein in the neonatal heart, we asked if we exogenously administer IL-13 following an MI in adults, can we improve outcomes? So we just took wild type C57 black mice and subjected them to myocardial infarction. And then we just performed a daily IP injection of either IL-13 or PBS for the duration of the 28-day experiment. And lo and behold, we found that when we exogenously deliver IL-13, when we look at cardiac function, we see a significant improvement post-MI. So this is fractional shortening that I'm showing you here. And what's important, our baseline and 3-day post-injury show that all animals are getting a pretty comparable sized injury, at least as assessed by 3-day post-injury echo. But the recovery from 3 to 28 days is significantly better when we give IL-13. Now, when we looked at the scars, we saw no difference in scar size. So at the time, I'm showing you this negative data because it becomes important later on in the presentation. But at the time, what this is suggesting to us is that we're getting sort of like an improved function of that remaining myocardial tissue. But IL-13 administration alone is not really significantly remodeling or resolving the scars. This improved cardiac function was associated with increased capillary density after MI. Now, we haven't really followed this phenotype up much at all, but it's interesting because this is kind of consistent. We've done these experiments in the global receptor knockouts, and we see that the capillary size and density is down. So this is at this point just like an associated phenotype, but it's consistently modulated when we activate or inhibit the pathway. So of course, in the neonates, I showed that we've at least demonstrated IL-13 signaling to myocytes and macrophages are both important. So we wanted to do the same in adults, determine which cell type is responding to IL-13 to improve cardiac function. So I'm not showing the cardiomyocyte data because it was negative. So when we gave IL-13 to mice lacking the receptor in myocytes, we could see some phenotypes, like they increased like EDU incorporation, suggesting there was some signaling event that was happening there, but we saw no functional improvement. They looked just like the wild type mice. So we moved on then to the macrophages. So we repeated these MI studies in the IL-4RA MAC knockouts. So they're missing the IL-4RA subunits, specifically in macrophages. And again, repeated our MI and daily IL-13 administration protocol. And what we found was that in animals of both genotypes, either the controls or the IL-4RA MAC knockouts, that PBS-treated animals showed a similar decline in fractional shortening. So this is, what I'm showing you here is the fractional shortening from 3 days to 28 days. So they're both genotypes show a similar % decline in fractional shortening. Now, when we gave animals IL-13, we again, just like we saw in wild type animals, we see this improved fractional shortening. But when we delete the receptor in macrophages, we don't see any improvement at all. We see a significant decline, similar to that, maybe even worse, compared to the PBS treated group. So this is really demonstrating that IL-13 signals directly to macrophages to improve the cardiac function post-MI. So this is where that scar data becomes relevant. We, of course, collected our tissues at 28 days post-injury. And as we saw before in just black 6 mice, which are essentially analogous to the control mice here, giving IL-13 didn't cause any difference in the scar size. We would have maybe expected it to be improved in mice getting IL-13 because they'd improved cardiac function. But when we knocked IL-4RA out in macrophages, now giving IL-13 doesn't only worsen cardiac function, but the scars are like 50% bigger than any other group, which it took us a minute to like kind of wrap our heads around that for a minute. IL-13 is supposed to be this beneficial cytokine. Now when we give it, we're getting worsening scars. So what's going on there? So what we think is happening, and this is ongoing work that I'm going to sort of summarize in a few slides, is that IL-13 can signal to fibroblasts or myofibroblasts. So we know the receptor is there. And that signaling may be causing a proliferation or expansion of this fibroblast population. Now, when we have IL-13 also signaling to macrophages, there's some interplay between those cell types that's suppressing that exacerbated myofibroblast expansion. So those are ongoing experiments that and we have some hypotheses as to what molecules are interacting between the two cell types. So in addition to exacerbated scarring, the IL-4A MAC knockouts that received interleukin-13 also showed a trend towards worsened survival. And so when we looked at the animals that were dying post-MI, this is just like the IL-13 treated animals. If we look at their cardiac function at 3 days post-injury, you can see some of the animals in the IL-4A MAC knockout group that had slightly better fractional shortening were still dying in the weeks following the injury. So what we think is if these animals had survived, we'd see these bigger, they have even more exacerbated scarring in this IL-4A treated MAC knockout animals. So our next question was, like, what are these macrophages looking like? What's the potential mechanism here? So we treated wild-type mice with IL-13 or PBS following MI, and then we collected immune cells for single-cell RNA-seq to characterize the composition of all immune cells, but I'm going to focus on the macrophages today. So we clustered macrophages into really 3 clusters. Our clustering actually found 5 clusters, but upon further analysis, 2 of them are actually dendritic cells. So we identified this MAC0 cluster, MAC1, and MAC3. And when we looked at what samples those macrophages are coming from, this MAC0 cluster of macrophages is coming from either IL-13 or PBS treated cells at 7 days post-injury. So these are kind of your, pro-reparative M2-like, if you will, macrophages. Now, if we look at the macrophages coming from the 4-day samples, what was really interesting is that we did see some macrophages from both the PBS and IL-13 treated groups in this MAC1 cluster. But this MAC3 cluster that's kind of an offshoot all on its own was derived from like 98% IL-13 treated animals at 4 days post-injury, so this early time point. So what was surprising about this at first to us was that we really didn't see much of a difference in the macrophage phenotypes at 7 days post-injury. Even when we ran like differential gene expression analysis, the macrophages at 7 days looked pretty similar between the PBS and IL-13 treated animals. It was really this early 4-day time point where we see these really distinct profiles. So to sort of conceptualize like what this means in the post-MI response, it's this really, classic description of macrophages that infiltrate the heart within the first, 1 to 3 or 4 days post-injury. We get this infiltration of these pro-inflammatory macrophages, which we can kind of consider our MAC1 cluster. And this MAC0 cluster, sort of this later time point, 7 days post-injury and beyond. These anti-inflammatory, alternatively activated pro-reparative macrophages. So what IL-13 stimulation is doing is it's really causing a shift in the phenotype of these phase 1 macrophages. Okay, so how do we identify these? I'll get to the characterization of what these macrophages are expressing and doing in a minute, but we wanted to be able to find these macrophages in vivo, okay? Because these are a really distinct phenotype. And if you go back to this, this plot here, these MAC3 macrophages kind of don't really exist in, you know, in the PBS treated heart. They're really distinct. So we wanted to be able to identify them in vivo. And we found that these MAC3 macrophages strongly and very specifically express IL-1R2. This, it's a neutralizing antibody for IL-1, and we can detect it as on the cell surface of these macrophages. What was really interesting is these IL-13 stimulated macrophages didn't really express like surface expression of CD206, which is kind of the classical marker of an IL-13 stimulated macrophage, which really demonstrates that the phenotypes that that are like the phenotypes downstream of IL-13 are really context specific. So what we know about IL-13 signaling in vitro really didn't translate that well to what we're seeing in vivo. So we developed an assay to be able to identify these IL-1R2 macrophages, and we were able to do that in the adult. So what we found was that compared to PBS, we get a significant increase in IL-1R2 macrophages with IL-13 administration. And when we delete the receptor in macrophages, that pretty much completely goes away. So this is a faithful marker of this MAC3 population in vivo. We then looked in the neonates, and compared to our FMO control, we already see that just in not like a non-stimulated, in just like a wild type animal, we already see more of these IL-1R2 macrophages compared to the adult. And sure enough, they pretty much completely go away in IL-4RA MAC knockout. So but basically, we're able to identify these macrophages in vivo, which is important for current experiments that we have doing some CRISPR screening of libraries to try to identify intermediates that can polarize or activate these. Mm-hmm. Mm-hmm.
There's enough cross-reactivity that it can compensate for interleukin 4. Right.
Mm-hmm.
We think of them as sort of being on the same team.
Yeah, they have a lot of overlapping functions via the type 2 receptor. Yes. Yeah.
If you give interleukin 13,
yep. Mm-hmm.
You get greater performance, but no change in scar. But if you take out the receptor, essentially, right, then you get an increase in scar.
Right.
And so you're probably going to get to this, and I just want to ask you, because I believe, what is it that, where do you get the benefits if you're not changing the scar size?
Where, like, how is it that, yeah. Right. So I sort of will kind of, so what I think is happening is the remote fibrosis is improved, and we're getting better contractility of the remaining cardiomyocytes. And what we have for sort of like the closest mechanistic insight is this efferocytosis that we have, this efferocytosis phenotype that we, yeah. But it's still not a definitive question. Like we don't know if like the contractility of the myocyte itself is better. It may be, but we're not there yet. Mm-hmm. Yeah, that's a question we're currently pursuing though. Right, so this gets to your question of like, what are these macrophages doing and how are they improving cardiac function? So we ran differential expression analysis specifically on the macrophages from 4 days post-injury and compared the macrophages in the different clusters from the PBS group and the IL-13 treated group. And as you might expect, we found that these MAC3 macrophages really completely shut off these pro-inflammatory programs. So even compared to MAC0 macrophages, these MAC3 macrophages have a significant downregulation of interferon signaling, for example. They also upregulate some matrix remodeling proteins like MMPs, and interestingly, upregulate this phagosome-related genes. So we hypothesized that these early macrophages have this enhanced capacity. So based on this increased expression of phagosome-related genes, this increased capacity to clear necrotic debris, so necrotic cardiomyocytes, for example, as this pro-resolving mechanism. So the first experiment we did to address this question was we performed MI, and then we, again, injected animals with IL-13 or PBS, and we administered these pH roto-green E. coli bioparticles. So they're essentially these, you just administer them intravenously, and when they're taken up by a macrophage, they fluoresce green in the acidic phagosome. So you can identify macrophages that are basically have this increased phagocytosis by flow cytometry. And what we found was that both in infiltrating or CCR2 positive bone marrow-derived macrophages, as well as in tissue-resident macrophages, we see a significant increase in these pH roto-green bioparticle uptake. So IL-13 is stimulating this basically uptake of these bioparticles. Now, uptake of these bioparticles is not really incredibly relevant, because what we're interested in is more of an efferocytotic pathway instead of a phagocytic pathway. So we repeated this experiment to specifically ask if IL-13 stimulated uptake of necrotic cardiomyocytes post-infarct. We developed this genetic mouse line that is a MYH6 Cree crossed with a lock-stop TD tomato. So all cardiomyocytes are fluorescently labeled red. So this is just our background strain. And then we repeated the MI experiment, and this time what we were looking for is macrophages that were TD tomato positive, implicating that they have taken up this cardiomyocyte debris. So when we repeated this, when we performed the MI experiment and we gave IL-13 or PBS, we found that IL-13 significantly increased efferocytosis, specifically of cardiomyocytes. And this was specific to the tissue-resident macrophages. So this is suggesting that IL-13 signals to resident macrophages. Those resident macrophages have improved capacity to clear this necrotic debris, which in other instances has been shown to be a pro-reparative mechanism following MI. Now, what was interesting, so we published this, and something that always was not really totally makes sense to me is that what's going on with these infiltrating macrophages? So we see this significant increase in the uptake of these pH roto-green particles in the infiltrating macrophages, but we don't see them taking up the cardiomyocytes. It's really the resident macrophages doing that. This kind of makes sense because resident macrophages during homeostasis, one of their roles is cleaning up necrotic and apoptotic cardiomyocyte debris. So that's a known homeostatic role for these resident macrophages. But we kind of wondered if these infiltrating macrophages are doing something or efferocytosing something besides myocytes.
Yeah.
Mm-hmm.
So the distinction between the infiltrated macrophages and the rest of the one
Yeah.
Possibly the infiltrating macrophages, ultimately becoming the rest of it.
Yeah.
Change the structure.
So, right, like if our flow cytometry, because we didn't do fate mapping for these experiments. So it's possible. So the markers that we're using are TIMD4 and CCR2. So those are two, fairly, especially with the addition of TIMD4 as a marker, most of them will be correctly assigned to like infiltrating or resident. But it certainly is possible, when macrophages infiltrate tissue, they turn off CCR2. TIMD4 is like double labeling both of those is a stronger indication of their origin, but certainly some of that could be happening.
Yeah. So, for example, if you kind of compare for the same animal, compare the spleen to what you're seeing there,
right?
Tell identity.
Yeah.
And say unequivocally that they really rest supposed to. Right.
Yeah, we could. Although they have to be, I mean, for this type of assay in particular, they have to be in the heart to be efferocytosing the cardiomyocytes. But yes, that's a good point. So correct. But we do think that these, another reason that we think we're missing something with these infiltrating macrophages is because when we look at the MAC3 population at the single cell level, those are predominantly CCR2 positives. So that, along with this finding, is suggesting that there's something that these infiltrating macrophages are doing that we haven't captured fully with this efferocytosis experiment. So those are experiments that are currently ongoing. So other evidence that we might be, that this efferocytotic phenotype might not be just specific to myocytes comes from this spatial transcriptomic experiment that we've recently done, where we specifically are looking at MERTK, which is an efferocytotic marker, and other markers show a similar profile. In wild type mice compared to PBS, we know that we get this increase in efferocytosis. And you can kind of appreciate, it's really hard to see here, that this sorry, let me just orient you a little bit better. These green dots, the bigger the dot means the higher the expression of MERTK. So you can kind of appreciate that in the remote region, I should have blown this one up as well, but you can see increased size of these green dots in the remote region compared to a PBS treated animal. But what was really striking is that most of these MERTK positive macrophages are actually in the scar region in the border zone and not adjacent to cardiomyocytes, but rather adjacent to other immune cell types and fibroblasts. So as we might expect, so this is just increased blow up of this border zone region. And you can really appreciate that compared to a PBS treated animal, we see a lot more expression of MERTK positive macrophages that are localized to the scar region. So we think that one of the perhaps mechanisms by which macrophages are interacting with the myofibroblasts is perhaps via some sort of efferocytotic mechanism. So that's a hypothesis at this point, but something that we're actively pursuing. This spatial transcriptomic experiment also demonstrated that indeed, when we delete IL-4RA and still give IL-13, that these MERTK positive macrophages are significantly suppressed. So they basically go away as we would expect if it's a direct signaling event. Okay, I just have a couple more slides here. So I sort of alluded to this over the last few slides. What we're really interested in, and some of the active CRELOX models that we are currently pursuing, are really understanding or proving that IL-13 is signaling to the myofibroblasts directly to exacerbate this scarring phenotype. So we've created single and double knockout models. So what to summarize kind of the work here, we know that when we give IL-13 and we hypothesize it can signal to both cell types, we get this improved cardiac function, no change in scar size. When we take the receptor away from macrophages, we hypothesize that IL-13 can then signal only to myofibroblasts, and it's lost this sort of inhibitory signal from macrophages. And when allowed to only signal to myofibroblasts, we get this unchecked proliferation or expansion of the myofibroblasts. So we predict that if we delete the receptor in both the macrophage and the myofibroblasts, we'll shrink that scar size back down. And then potentially, if we delete the receptor from the myofibroblasts and only allow this macrophage signaling, maybe we'll get even improved function over baseline. But these experiments are currently in progress. So to summarize the mouse work that we've done, we've shown that IL-13 and or IL-4 signaling to neonatal macrophages, I haven't depicted here the cardiomyocyte story because it wasn't much of a focus, improves cardiac function and scar resolution. When we exogenously administer IL-13 can signal directly to macrophages to increase phagocytic or efferocytotic capacity. It increases matrix remodeling genes, and these cells can be identified by IL-1 or 2 surface expression. And this collectively results in increased function and decreased exacerbation of scarring. Now, I just wanted to end with this clinical perspective. So we often wonder if, the pathways that we're studying in mice have anything to do with human heart failure. So my very talented graduate student, as like one of the last parts of his dissertation, was to query this collaborative patient data network called Trinetics. And what he looked for was how patients respond when they're on this drug called dupilumab. So dupilumab is an IL-4RA neutralizing antibody, and it is used
in the way everywhere
to treat atopic dermatitis, like severe cases of atopic dermatitis. And this drug was it was approved in 2017, so relatively recently. And so what Santiago did was he queried this massive database and identified individuals who had an index event of a cardiac ischemic event. So either MI or an unstable angina or something like that, without heart failure. So they were not diagnosed with heart failure within the first 3 months. So this is important because we're kind of stratifying for patients that have a modest size MI, not something that has completely destroyed the heart and their immediate heart failure. So we perform propensity score matching on every piece of data available to us, including any drugs they were on, sex, demographics, age, any pre-existing condition. And also importantly, we matched for patients who had the same atopic dermatitis condition. They're the same underlying condition, but some were prescribed dupilumab and some were not. And what we found was that over the course of, we took this out to 1,500 days, and this time point was limited by how long the drug has been approved. And what we found was that patients who were on dupilumab, shown in red here, had an increased incidence of developing heart failure over the 1,500 days compared to patients who were not on dupilumab. Now, of course, this could be confounded by perhaps severity of the underlying condition or other confounding factors that we don't have data on. But this suggests, along with our studies in mice, and others have also published pro-reparative or beneficial effects of IL-4, IL-13 signaling, that this could be a real therapeutic target modulating this pathway for treating heart failure in humans. So with that, I'd just like to acknowledge the members of my lab, particularly Santiago Alvarez, who did a lot of this work, and he's now a postdoc at Northwestern, actually in the Art Holly lab, coincidentally. And my collaborators in department and of course our funding sources. And if there are any other questions, I'll be happy to take them. Mhm.
Yes.
Yep, thank you.
Last, it's an illustration.
Mhm.
So with the, deletions,
Mhm.
the application, is that a global thing or this specific?
It is global, so it's with the CX3